we're here now. Say you had a time machine and you zoomed forward in time, assuming the Earth was still around at this point. Fast forward 100 million years. What do you think life would look like?
So if we fast forwarded 100 million years from now,
Yeah.
well, I think that's actually a good question because, based on mutation accumulation, right? I mean, we know we inherit about 100 new mutations per person per generation. And based on the known functionality of the genome, most of those are deleterious. So if I were to look 100 million years into the future, I would see extinction, because that's exactly where we are heading, since there's no type of selection that can remove all these deleterious mutations.
You said there that most mutations are deleterious.
Yes.
That's not true.
Well, it is true, because
most of them are neutral.
No, well, I mean, according to the assumptions of evolution, most would be neutral, but that's just because evolutionists assume that the majority of our genome is based on evolutionary leftovers, junk DNA, genomic fossils, but that's actually a direct prediction of the created heterozygosity hypothesis that would suggest that the vast majority of our DNA elements, for example, ERVs, ALUs, and these other classes of retrotransposons would be functional. And that's exactly what we're now seeing. That means the more functional the genome is, Adam.
Can I just ask quickly there? Did they make that prediction before we found that out?
Yes, intelligent design advocates have always predicted genome function increases in levels of genome function. That's why the evolutionist community attacks the findings of the ENCODE project, because Adam, the ENCODE project has shown that most of the human genome is functional, and not only that, but functional on many levels. I mean, we have layer upon layer upon layer of programming within the genome. And are you familiar with all the known functions in these retrotransposons and in the endogenous retroviruses and pseudogenes?
I'm not familiar with every single one, no.
So Dr. Nathaniel Jensen, in his model of created heterozygosity, and that's why I asked you to clarify your understanding of where we explain the origin of the vast majority of genetic diversity, for example, in the nuclear DNA, because it's two opposite expectations and predictions. I mean, how much of our genome on a percentage wise, both protein coding and non-protein coding, would you say is functional according to your model?
According to my model, I don't know. I don't know the number, unfortunately.
It is very low. Like I've debated PhDs on this, and they have to assume junk, for one, in order to counterbalance the mutations that are building up in the genome, because they need to assume that they are neutral, that they're not actually damaging the genome. But even if, and it's funny, because the evidence suggests that we have a genome of function, but even if, just for sake of argument, just to not discuss this topic forever, even if I said, okay, let's pretend that the genome really is 90% junk, that still means that's 10 new deleterious mutations that are pouring into the genome per person per generation, and most of those being unselectable, because natural selection, Adam, only acts on the most detrimental of mutations and the most deleterious mutations, but it does nothing against the nearly neutral mutations. Those are invisible to selection. So my answer to your question, if we were to go 100 million years into the future, it's a very simple question. I mean, life would not exist. And I guess you would have to explain how natural selection can keep life in existence for that long. Go ahead.
Yeah, you talked about genetic entropy. Now, I looked up genetic entropy, and the only places I could find it on Google Scholar were creationist sources, specifically from John Sanford. And all the examples I see are very specific, species-specific examples, mostly humans. Now, the problem with humans in the natural world is that we've sort of done away with natural selection. So, yeah, deleterious mutations are more numerous than beneficial ones. So over time, if there is no selection pressure, you'll have more of a buildup of deleterious mutations. However, natural selection in the wild, assuming that it's able to do what it needs to do, will prune the populations of deleterious mutations and keep the positive ones. So that's how you maintain populations. That's what natural selection does. It gets rid of the bad and it keeps the good, what's fit for the environment that the creature is living in. That's how populations are sustained, and that's how they have been sustained.
Right. I really appreciate that response, because you made some good points. I just don't think that you've addressed the nearly neutral mutations that would build up, because the same thing goes, and I don't want to take all the time. I know Matt has some good examples of genome degradation, and health, for example, is kind of his specialty. So I'll let him take over in 1 second. But I do want to point out the fact that, in humans, so obviously you're admitting that we are degenerating, but you are saying that it's due to a lack of natural selection, right? Which makes sense. But it would come down to, let's say, animal populations. It would still come down to genome function as a whole, because even in animal populations, they still have to assume that the majority of the genome is junk in order that the junk areas can absorb the mutations, making them neutral. But the fact is, Adam, and you have to address it, is the evolutionist has lost one of their favorite pieces of evidence, which is not only junk DNA, but also the presence of these ancient deactivated viruses that they say are in the genome. Because rather than being functionless vestigial remnants of our past, these retrotransposons, as a direct prediction from our model, turn out to be functionally integrated into the amazingly complex regulatory genetics and genomes of mammalian genomes. So that means that total functionality with all these DNA elements, DNA sequences, will speed up and make the degeneration problem that much worse in humans and in animals. Take your time.
I am very curious, because you keep saying that intelligent design predicted. Who predicted it and when?
So there have been predictions in the past. I can get you specific names. For example, Dr. Jonathan Wells has made those types of predictions. Dr. Nathaniel Jensen has in print right now new predictions that the vast majority, over 80% of our genome, because ENCODE, they did what's called biochemical tests, right? So Dr. Nathaniel Jensen predicts that using genetic knockout tests will reveal exactly what types of functions these DNA differences have. Because if we predict that the vast majority of DNA differences are actually functional, Adam, that would be evidence for that exact hypothesis. We're seeing that with the overturning of the junk DNA. I mean, what was called junk DNA, we now know can modify the way chromosomes, right, the packages of DNA, are organized. And then what that does is it changes the way the DNA functions. It's DNA function on multiple levels. I mean, how do you address all that?
Non-coding areas of the genome do exist in large quantities. And in fact, it's one of the ways that you get de novo genes, which explains orphan genes. So, for example, a study recently looked at cress, several species of cress. And they found that these lineage-specific genes, or what they call orphan genes, were actually just mutations of non-coding DNA that didn't do anything in other species of cress.
Right, and that's the assumption. So
no, it's not an assumption. They tested it.
Well, no, I understand that, the explanation for these taxonomically restricted and essential genes is de novo gene synthesis. I'll address that in a second, but I wanted to point out that if the genome truly is junk in the non-coding region, most of it, has no active role. But then the ENCODE project has revealed that over 80% of it is actively transcribed into RNA, and the evolutionists say, well, that's just biochemical testing. But my question to you would be, if it truly was junk and there was no useful activity there going on in the non-coding regions, why, Adam, would the cell even bother with it? Because wouldn't you agree that activity that is discovered would be just a waste of energy and resources, and natural selection, which we were talking about earlier, should have eliminated that junk long ago, according to evolution? How do you answer that?
I'd have to look into it more. I couldn't answer off the top of my head without sounding like a complete dunce.
No, that's okay. I'm enjoying it. I do want to pass on to Matt real quick, because his expertise on health and genetic degeneration, I wanted him to at least point out a few
I would
examples.
like to just interject something here. I would like to eventually get back to kinds, because we keep drifting back to evolution stuff, and your idea is that disprove evolution.
Well, no, it's not disproving evolution because, and I know that the assumptions and basic worldviews of evolution, it's hard to, put themselves into the mindset of a creationist, because what we're saying is that the vast majority of DNA differences were the result of divine creation. An evolutionist says it's mutations over time. That means we have two very different expectations and predictions on ancestry. You would expect, a genome of junk, evolutionary leftovers, and genomic fossils. We would expect a genome of function and DNA elements. That's what's going to help us determine whether or not everything's related, or whether or not limited ancestry, according to the Bible, is what's true. And that's exactly what we're pointing out here and demonstrating.
Okay.
Is that
So what could you explain then why every creature, if you test them genetically, comes up into this well-defined tree?
Right, and that's a good question. You're going back to the nested hierarchical patterns, the groups within groups patterns that we see in life. And as I pointed out in the beginning, the design model predicts and expects the exact same thing. That's why we have to look at differentiating evidence, which would be function. How do we test, for ancestry? It would be function, a genome of function versus a genome of junk. But like you said, let's go more into kinds and more expectations of speciation from, say, the original kinds, because I know that's what you want to talk about, and I think that would be enjoyable for the audience. But can we just reserve just 1 second for Matt to jump in and make a point about genetic entropy, and then we'll go right to kinds and definition of kinds and things like that?
Yeah, that's absolutely fine with me.
All right, I'll just get it over with real quick. A perfect example is a video that I made, which shows Neanderthal. It shows that they have less overall genetic mutations in their entire genome, and today we have a lot more. We also have something called the MTHFR gene, and it's broken anywhere between 40 to 70% in almost all people on Earth. And it's that gene determines on how well you methylate and how well your other genes work and how good they are. So it kind of shows a genome collapse by looking at that. But that doesn't really have much to do with kinds. It's just an overview of showing that the genome is kind of crumbling and breaking down. We see gene loss. We don't see new genes arising and our genome going up. We see it going down in all life. So
I will just put a point on that. We do see genes arising. I mentioned the CRESS earlier, where they identified many non-coding sequences becoming de novo genes. So we do see increases in genes. We also see anatomical increases, which are results of genes. So for instance, dogs, more primitive dogs, wolves and huskies, don't have puppy dog eyes because they lack two muscles around the eyes, whereas more recent breeds of dogs have these muscles, and that is a result of mutations that came about to benefit dogs that live with us because we love puppy dog eyes. I think that's a beautiful example of evolution in action that happened during human history.
But these de novo genes that are arising spontaneously, kind of like out of nowhere, that really are considered junk DNA, are actually functional elements. So we can't just say that these are randomly arising, but yet these de novo genes are absolutely required for functions in the gene in the body. If they were just, that's what makes the orphan genes so fascinating, is because they're lineage specific. So it also helps prove our model a little bit, because they are taxonomically restrictive genes, but they're also functional elements, which Standing mentioned earlier.
The fact that genes become active from non-coding genes, I don't see how it proves your point. I don't see how it lines up with it.
And I like your point on orphan genes, Adam, but we would just have to see actual, papers that are showing a non-coding region of our genome suddenly popping up and having an incredibly functional role. Because
if you look at the one on CRESS, it was quite recent. If you look up CRESS de novo genes on Google Scholar over the last, I think it was 2016. If you look at it, there's about 500 de novo genes that come up from non-coding regions.
And we have looked at many papers. I just want to put this out there, is that in what you're saying, just to reiterate, these orphan genes were created supposedly from just random mutational events in the non-coding regions, and they've now co-opted function in the protein coding regions. They're not, right? Is that how you would explain the orphan genes?
We see the non-coding regions. The reason we didn't see them before is because they were non-coding, so we didn't really know what we're looking for. But when we found these genes in this certain species of cress, we looked back at other species of cress and checked for that same sequence and find that it's a non-coding region of the DNA.
Well,
I'm saying cress. I'm assuming you think cress would be within the same kind.
Right. Well, and I'm just saying, I'll give you plenty of time. I'm just saying that because evolutionists reject this concept of engineered design in biological systems, when they see the presence of these taxonomically restricted orphan genes, they have to come up with a way as to how these cleverly, designed DNA sequences were somehow randomly generated in very recent evolutionary time, and you look to de novo gene synthesis. But my response to that would be that it's based more on a circular argument, because evolutionists say, okay, de novo gene synthesis must be true because why? Orphan genes exist, and orphan genes exist because of de novo gene synthesis. So I don't know how
there are various methods for not de novo genes, orphan genes. You've also got things like fission and fusion of genes. You've got gene duplication and exon shuffling off the top of my head.
Right. But a lot of times in these papers, for example, one that Matt and myself are really familiar with are the so-called introduction of new orphan genes in fruit flies. Matt, did you want to touch on that real quick? Because orphan genes, you must understand, for one, could be lost as well, which could explain why you're seeing some in some species in the same kind that other species in the same kind don't have. Did you want to touch on that real quick, Matt?
Yeah, real quick. They took fruit flies and they looked inside of their orphan gene status, and they wanted to try to get them out. So what they did is they bred fruit flies, a specific species of them, to have 0 orphan genes in their bodies, just to see what would happen. So they bred them entirely out after maybe 10 generations or so, completely gone. So the flies were living without any orphan genes in their body.
Okay, I just want to make a correction. I said 2016 for the cress paper. It's actually 2011. So if you wanted to search on it, it's a paper from 2011.